I'm Dr. Nathaniel Chin, and you're listening to Dementia Matters, a podcast about Alzheimer's disease. Dementia Matters is a production of the Wisconsin Alzheimer's Disease Research Center. Our goal is to educate listeners on the latest news in Alzheimer's disease research and caregiver strategies. Thanks for joining us. Welcome back to Dementia Matters. I'm joined again by Dr. Reisa Sperling for the second part of a 2-episode series. If you haven't listened to part 1 yet, Dr. Sperling is a neurologist and leader in Alzheimer's disease research. She holds numerous titles and roles, including serving as a professor of neurology at Harvard Medical School, the director of the Center for Alzheimer's Research and Treatment at Brigham and Women's Hospital and Massachusetts General Hospital, co-principal investigator of the NIH-funded Harvard Aging Brain Study, and one of the principal investigators of the Alzheimer's Clinical Trial Consortium. She is also a leader in clinical trials, having co-led the A4 study, the first prevention trial in late-onset preclinical Alzheimer's, and the AHEAD study, a trial testing whether lecanemab could lower a person's risk of memory loss due to Alzheimer's. In the first episode, we discuss what preclinical Alzheimer's is and the roles amyloid and tau play in cognitive decline. If you haven't listened to that interview yet, you can find the link in the episode description. On today's episode, we're going to dive a little deeper into the different factors that can impact cognitive decline in early-stage Alzheimer's disease and how clinical trials are shaping the field's understanding of detecting, treating, and preventing the disease. Welcome back, Dr. Reisa Sperling. What are the key factors that increase the risk of cognitive decline in Alzheimer's disease? And how do gender and the presence of ApoE4 gene play a role in that decline?
The studies so far really suggest that the biggest risk for decline is amyloid, and in particular, markers of amyloid that also tell you something about tau. So blood-based biomarkers of phospho-tau is actually far and away the strongest predictor of cognitive decline. Having said that, though, I think that we have evidence from several studies, and we're now starting to look at this in the A4 study, that amyloid plus other processes, in particular vascular risk, so just the Framingham risk score, high systolic blood pressure, or other vascular risk, seems to interact with amyloid and increase the amount of tau you have for a given amount of amyloid and increase your risk of decline. There's also evidence that this vascular disease interacts with amyloid in causing other proteins. There's one people are very interested in called TDP43, or sometimes called late disease, that also seems to be related to vascular risk. And interestingly, you brought up ApoE4. Also, ApoE4 is a risk for both Alzheimer's disease amyloid as well as for TDP43. So I think ApoE4, its primary role seems to be that it increases the amount of amyloid you have for a certain age. I would say the jury is still out about whether E4 carriers have an increased risk of decline for the amount of amyloid they have in their brain. There is some evidence of that, and particularly in female ApoE4 carriers, not so much in male ApoE4 carriers. But again, I think ApoE's main risk factor is by increasing the likelihood that you'll accumulate amyloid at a younger age.
And then you did mention that protein TDP43, but I know our listeners are also sending questions asking about Lewy body proteins and alpha-synuclein Parkinson's. Maybe we don't have enough of this data, but do you see individuals having different rates of decline the more brain diseases that they have?
Yes, there are some really important studies suggesting that people who have both Alzheimer's pathology and Lewy body pathology unfortunately have the fastest rates of decline. And we think of these as very separate diseases, but in fact, almost all patients with Alzheimer's disease have some Lewy bodies in certain parts of their brain, even autosomal dominant, these dominantly inherited Alzheimer's disease young people. Almost all of them have Lewy bodies in this small part of the brain called the amygdala. And no one fully knows why, but nearly 100% have Lewy bodies in their amygdala. And more than 50% of people with Lewy body dementia have at least amyloid, although they don't all have tau. So, again, to me, I think of them as proteins that accumulate in our brain as we get older. We unfortunately have a hard time emptying that protein garbage. And why some people get more Lewy bodies and other people get more amyloid and tau is another unclear question, but I think the processes that underlie them have some similarities.
Now, how do cognitive baseline scores and then the rates of actual decline differ between men and women in Alzheimer's disease? And if you could go back to even including those with the ApoE4 gene.
So women tend to perform better at baseline on these tests that we have, and that's because they're mostly verbal memory tests, and I don't think anyone's going to argue that women might be more comfortable talking. So these verbal memory tests, again, almost all of them, even when we try to equate them, women perform better at baseline. But a number of studies have suggested that women decline faster than men. So, one possibility, again, is they start at a higher level, so they have farther to fall. But more recent data also suggests that there may be biologic reasons for this. So, women who have elevated amyloid in their brains, they get a little more tau for a given amount of amyloid than men do. But it's an interesting paradox because men have more vascular risk factors. And so you would think that if vascular interacts with amyloid for tau, that men should have faster decline. And that's where this ApoE4 comes in, because there's something about women with E4 alleles that increases their risk of decline, especially on these verbal memory measures. And this was noted actually way back when ApoE4 was discovered. This is now 35 years ago, I think, that the effects were really driven by women. And we still don't fully understand that sex by ApoE interaction. And I think that's going to be continued work.
Now, Risa, you've spent a good amount of time also bringing up vascular disease, and I'm hoping you can comment with a little bit more detail now about how vascular disease in the presence of amyloid or in the presence of tau contributes to the disease process of Alzheimer's to cognitive decline. But if you could start by just even how do you define or how do you think about vascular disease? You mentioned blood pressure, but are there other factors that you think about that are going to be meaningful, both as a scientist, but then also as a physician?
Yes. I think that vascular disease, or more specifically here, I'm talking about changes in the blood vessels in the brain. Here, although they often are partners with blood vessels in the heart and the rest of the body. I don't think you have to have a frank stroke to have vascular disease play a role in risk for cognitive decline. There's been a number of studies, including from the vascular researchers like the Sprint Mind study, as well as studies that are looking at risk for Alzheimer's disease that implicate what we call vascular risk as playing a major role here. And in particular, the two that have stood out in our studies are blood pressure, both systolic blood pressure in some studies and what we call mean arterial pressure, which is, the average of the top number and the bottom number of your blood pressure, contribute to the risk of decline. And especially now we can see that that's important because it's associated with faster tau accumulation. And then the body mass index, or BMI, that we all talk about, has a kind of interesting piece here. So, overall, unfortunately, obesity is associated with vascular risk. And we've known that the Framingham Heart Study has a risk score, and their BMI is a major risk for future vascular events, a heart attack, a stroke, and for dementia. But interestingly, when you look at people who are beginning to accumulate amyloid, they lose weight. And so we always try to get people to lose weight in midlife, especially. But when somebody who's older starts to lose weight, especially if they have cognitive problems, that's often an early sign of late preclinical Alzheimer's disease.
So, not necessarily that the losing of weight is contributing to the pathology, but that it's actually a marker that the pathology is starting to take effect.
Exactly. So, especially again, somebody in midlife being overweight or obese increases their risk of late life cognitive decline. But for some reason that we don't understand, the buildup of amyloid is associated with weight loss in late life, and that's not a good thing once you're over about 70 or so. You also asked me how I approach this as a clinician. And I will say that I think part of the reason we've also had some success in epidemiologic studies where we can see that the rate of rise of dementia is actually slowing down. So we're not, unfortunately, having a lot less dementia because people are living longer and that increases the risk. But the rate of rise is going down, and I do attribute that to public health differences in controlling blood pressure, controlling cholesterol with better diet, and also with statins. And I think these vascular interventions have made a big difference in the percentage of people who progress to dementia. Vascular risk does not seem to affect amyloid much, but it may affect tau, but it also may change the ability of the brain to be resilient. So it may be that part of what vascular disease is, even if you haven't had a stroke, but you have small, what we call small vessel or white matter hyperintensities on an MRI image, or just making the nerve cells less resilient to other insults that may come its way. I definitely approach vascular risk when I see patients and say, while we're trying to help your Alzheimer's disease, let's maximize your vascular protections.
I want to transition to some of the work you do in the clinical trial space. And you said some very interesting things at presentations about primary prevention. And so, what does primary prevention of Alzheimer's disease look like, especially when you're considering and talking about a concept of preclinical Alzheimer's disease?
Yes. So the most exciting thing for me right now is partly that I can see a future where we try to prevent the amyloid plaque buildup and the tangles from building up altogether. And that for me would be primary prevention. The trials we do now, I refer to as secondary prevention because they're in people who already have amyloid plaque buildup in their brain and often have tau tangles starting to build up. And our job there is to prevent the cognitive decline. But ultimately, we want to prevent the brain changes to begin with, and that's primary prevention. And with the advent of these blood tests that can tell us first, right now, whether things are just starting to be abnormal, even before you can see the amyloid PET buildup, then that can get us closer and closer to primary prevention. And I can imagine a day where we treat everybody either at age 50 with a vaccine or something that will decrease their likelihood of buildup, or perhaps we start with people at high risk, either because they have ApoE4 or they have another family risk, and we try to, again, prevent that amyloid buildup from starting. And that we need to start early, probably age 50 or 55.
Now, you mentioned secondary prevention trials, and the one that we've talked about already together is the A4 study. And even though it didn't show treatment differences in cognitive decline, you have mentioned that there were a lot of benefits that you've learned since then. Could you just provide a brief highlight of some of the key takeaways that you've gotten from the A4 study?
I think the most important finding from the A4 study was looking at what the greatest predictors of decline were. And we first saw looking just at the levels of amyloid PET at baseline, and that top third of individuals, people who had a lot of amyloid, even though they started out normal and really pretty normal, they had a very high risk of decline on our memory test. And they also had almost a 50% risk of progression to mild cognitive impairment in 5 years. So that was really um, striking data that very high levels of amyloid are associated with very substantial risk. We also were able to go back and look at blood tests and found that the blood tests, especially this p-tau 217, was as predictive, even more predictive than just the amyloid PET rate. The other thing that I think was very important that we learned from the A4 study is that the relationship between this cognitive decline on our tests and the likelihood of progression using clinical functional tools like what we call the clinical dementia rating scale, that they actually track together. First, you have cognitive decline, and then often in the later years, you develop cognitive impairment. And that's really helpful for future trial designs so that we can, depending on how much pathology you start with, you can design a trial that will see change in a shorter time or a longer period. We're still looking at a lot of other data, though, including lots of genetic risk factors and looking at effects of race and ethnicity. The A4 study was not nearly as diverse as I wish it was, but we did learn from that there are differences in the rates of PET and plasma abnormalities by race and ethnicity that I think is very important and informs both the AHEAD study we're doing now as well as future studies.
Well, I did want to ask you about the AHEAD study, which I've been told is also A3 and A45, but the AHEAD study. And I was wondering how have advances in the plasma pre-screening, particularly with p-tau 217, which you've brought up, how has that improved the efficiency and accuracy of detecting early stage Alzheimer's disease?
Yes, so plasma became available, and we started to see that there were accurate tests in blood tests during the first year of screening in the AHEAD study. And we said, wow, this could really help our what we call a screen fail rate on amyloid PET. Because when we started the study, about 70 or 75% of individuals didn't show even the intermediate levels of amyloid on PET scan. And that's partly because the AHEAD study goes down to age 55. And the younger you are, the less likely you are to have a lot of amyloid plaque buildup. So we did the first study where we looked at the first 1,200 people we screened. At that time, the blood test mostly looked at amyloid called A beta and a ratio. And using that ratio, we could decrease our screen fail rate from 70 or 75% down to 55 or 60%. But a year later, phospho-tau 217 came along, and we were able to look at those measures and did a much better job at predicting how much amyloid was there. We then could actually get to close to 90% accuracy in predicting with a combination of the amyloid blood test and the phospho-tau blood test. And most recently, we now have about a 30% screen fail rate in AHEAD at the end of screening. So it's really, cut the need for amyloid PET screening in half or more. And I think in the future, we will see these blood tests used both to select people for clinical trials, and if these trials work, to select people who should get treatment, even when they have no symptoms apparent.
Well, your research and others in clinical trials showed that individuals with the lowest amyloid burden respond the best to treatments, particularly monoclonal antibody therapies. So, can you discuss the implications of this finding for future therapeutic approaches?
Absolutely. So, one of the very exciting things for me with both the lecanemab trial, the first fully FDA'd, and then very similar data from the donanemab trial that was recently approved. So, both of these trials only enrolled people who had at least mild cognitive impairment. They had some symptoms. But we were able to look at the group who came in with the lowest amyloid and especially the lowest tau and could see that the positive clinical response was seen and in some ways driven by these groups who had the lowest amount of pathology. Now, we were able to look at the group in the lecanemab who had a low tau PET, and that was the group, some of them even appeared to get better than their baseline or at least stay stable, but it was a pretty small group. So we also were able to look at people on an amyloid PET cutoff that corresponded to that low tau, and they also responded better. And then in donanemab, they had actually designed their trial to include tau PET, and they could see that their response was much stronger in the low to intermediate tau group. So for me, this was a clear pointing to the path forward, which is if we could go even earlier, as we're doing in the AHEAD study, and also with donanemab, people are testing in the Trailblazer ALZ-3 study, find people who were asymptomatic or very minimally, they had some cognitive concerns, but they're not yet impaired, who also have these low levels. Maybe we could prevent decline altogether. And so I'm very excited about the evidence pointing to clearly earlier is better. And I'm thrilled that we have trials doing that. You mentioned the A3 part of the AHEAD study, and I'm particularly excited about that because that's just a phase 2 study within AHEAD. It was at the time people thought, oh, you can't do this. So we looked at people who weren't even clearly amyloid positive yet. They had an intermediate range, which now people increasingly recognize as abnormal amyloid. But that group, we have amyloid and tau PET in everybody. And if we can show that we prevent the tau from spreading, then I think that's really strong evidence that we can get to prevention of cognitive decline in the future. So I'm very excited about the A3 piece of AHEAD in the future.
So, in our time together, we've talked about preclinical Alzheimer's disease, amyloid, tau, p-tau, additional risk factors for cognitive decline, and now prevention clinical trials. So, to end today, what excites you the most about the next 5 years?
I would say what excites me the most is that I do think we will have successful secondary prevention, meaning people who we can find the process in the brain and prevent them from developing impairment. I hope that both AHEAD and Trailblazer ALZ-3 are positive, but I think there's a fairly high likelihood in the next 5 years that we will get an answer there. But most excitingly, it's that within 5 years, we're going to start these primary prevention trials where we're really, I think, going to be able to go after the underlying changes and prevent them from building up. And this is exactly what happened in cardiovascular disease, right? We now treat people who have barely elevated cholesterol, and we put people on medications that keep their blood pressure quite low, trying to prevent the vascular disease from really getting started. And I can see that happening in Alzheimer's disease. I'm excited about vaccines, particularly in the future, or maybe pulse dosing of antibodies. I think we will, in 5 years from now, be really testing whether we can prevent brain amyloid and tau tangles altogether. And I can't wait.
Well, your excitement's gotten me excited, Risa. Thank you, and thank you for today for discussing all of this with us on the Dementia Matters podcast.
Oh, thank you so much for doing this, Nate. I really appreciate it.
Thank you for listening to Dementia Matters. Follow us on Apple Podcasts, Spotify, or wherever you listen, or tell your smart speaker to play the Dementia Matters podcast. Please rate us on your favorite podcast app. It helps other people find our show and lets us know how we're doing. If you enjoy our show and want to support our work, consider making a gift to the Dementia Matters Fund through the UW Initiative to End Alzheimer's. All donations go toward outreach and production. Donate at the link in the description. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center at the University of Wisconsin-Madison. It receives funding from private, university, state, and national sources, including a grant from the National Institutes on Aging for Alzheimer's Disease Research. This episode of Dementia Matters was produced by Amy Lambright-Murphy and Kaylin Rauerdink and edited by Alexia Spevacek. Our musical jingle is Cases to Rest by Blue Dot Sessions. To learn more about the Wisconsin Alzheimer's Disease Research Center, check out our website at adrc.wisc.edu. That's adrc.wisc.edu. And follow us on Facebook and Twitter. If you have any questions or comments, email us at dementiamatters@medicine.wisc.edu. Thanks for listening.